Hemostasis begins when you have endothelial cell injury. Injury of the endothelial cell causes the collagen that's underneath the endothelial cell in the vessel wall to be exposed, and von Willebrand's factor, from a variety of sources, including the endothelial cell itself, will bind to this collagen. When von Willebrand's factor binds to collagen, this complex is recognized by GP1B on a naive platelet. That's one of the receptors that's relevant for this first step, GP1B for the first step, for adhesion. Once the platelet has adhered to the vessel wall, it has an interesting change of shape. Along with this change of shape, the alpha granule within the platelet will start to produce a few things. It makes von Willebrand's factor again. It also produces fibrinogen, which is factor 1, and it also releases platelet factor 4. The dense granules within this activated platelet will also release ADP and calcium. Lastly, the cyclooxygenase enzyme within this activated platelet will create thromboxane A2, or TXA2 for short. Now, from this adhered platelet that has activated, how can we activate further platelets? Well, the ADP that was released from the dense granules will go ahead and bind onto other receptors on another platelet. These receptors are called the P2Y1 receptor and the P2Y12 receptor. Again, this is another naive platelet, that's why it's round in shape. When the P2Y1 receptor is bound by ADP, this causes calcium to come into this platelet, which is what actually causes this second step of activation. When the ADP binds P2Y12, this takes us to our third and final step of aggregation. Once several platelets have basically activated, and they all have the stellate shape, what holds them together is this GP2B3A receptor and that factor 1, or fibrinogen, that was released before. So GP2B3A and factor 1 are what cause aggregation. The thromboxane A2 also binds to its own receptor called the thromboxane receptor, and that also promotes aggregation. So adhesion, activation, and aggregation are the three major steps in primary hemostasis. To tie this to secondary hemostasis, we also need to recognize that the calcium ion that's released by platelets is extremely important in the activation of the coagulation cascade and the coagulation factors. And lastly, this factor 1 is released by platelets through their alpha granules, but it is also one of the coagulation cascade factors that can be further activated to fibrin. And then this fibrin, or fibrinogen, can be cross-linked via factor 13, which is what will take this current weak platelet plug and make it into a clot.